Hey everyone, welcome back. Today we're going to be talking about something pretty insane, something that will affect you if you're alive today. So scientists have reversed the signs of aging in monkeys. Humans and monkeys both belong to the primate group, and with some similar biology, this breakthrough is indeed exciting. Our adult stem cells play a crucial role in regenerating our lost or damaged tissues, rebuilding our body cell by cell. However, with age, our bodies become riddled with inflammation, hardly providing an environment capable of keeping our stem cells healthy. Eventually, our stem cells lose their regenerative capacity, contributing to degenerative aging. Hydra, a genus of tiny freshwater polyps, are considered biologically immortal because they don't age and can continuously regenerate damaged cells and tissues. However, they are not truly immortal, as they can still be killed by external factors like predation, starvation, or pollution. Their ability to live indefinitely stems from their constant production of stem cells that replace all their cells every few weeks. Hydra can escape death by infinitely regenerating. Their stem cells can continuously proliferate and renew by producing FOXO, a protein they share with humans. In simple terms, FOXO is a family of cellular supervisors that instruct your body's cells to perform beneficial and protective tasks, like repairing themselves, crucial for preventing aging. What this all means is that enhancing FOXO activity slows down aging by promoting cellular health, stress resistance, and stem cell functions. Scientists did an experiment to see if senescence-resistant stem cells, abbreviated as SRCs, could biologically slow down or even reverse the signs of aging. SRCs are genetically engineered cells designed to withstand the harsh environment of aging and resist cellular deterioration. To test this, an experiment was done on old monkeys. The oldest of the monkeys, the A4 group, were the focus of the study and were subdivided into three groups. One group was injected with saline, salt, and water, another group with normal stem cells, and the third with SRCs. The aged monkeys were injected every 2 weeks for 44 weeks, approximately equivalent to the duration of 3 human years. On safety, there were no serious adverse events, such as immune system rejection or tumor growth. After 44 weeks of bi-weekly injections, a suite of biological indices was measured from the aged monkeys to assess whether SRCs slow down biological aging. One of these indices was memory retention. To assess memory, the researchers conducted a common experiment called the Wisconsin General Test Apparatus, WGTA. For this experiment, each monkey was trained to retrieve food located outside of one of 2 identical boxes. During the test session, after each monkey was trained, food was placed next to one of the boxes to keep it hidden. Subsequently, a flap was placed in front of the monkey to block the boxes from view. 3 seconds later, when the flap was reopened, each monkey had to remember which of the 2 boxes contained the food. Remarkably, the monkeys injected with SRCs remembered the location of the food with higher accuracy than the monkeys injected with saline. Moreover, the monkeys injected with normal stem cells exhibited the same level of accuracy as the monkeys injected with saline. These findings suggest that SRCs, and not normal stem cells, improve the memory of aged monkeys. Furthermore, an MRI scan showed that treatment with SRCs decreased age-related brain shrinkage. MRI-based experiments also revealed that brain connectivity was restored to that of young, A1 group, monkey. Also, the structural connectivity between 7 brain regions, including those important for working memory, prefrontal cortex, was rejuvenated with SRC treatment. Overall, these findings suggest that SRC injections improve memory by protecting the nerve cells, neurons, in the brain and spinal cord. In addition to the brain, the Academy researchers found that SRC treatment rejuvenated multiple organs and tissues. This is important because the rejuvenation of a given organ or tissue could lead to the reduced risk of its corresponding age-related chronic diseases. For example, the rejuvenating effects of SRCs on the brain could reduce the risk of neurodegenerative disorders like Alzheimer's and Parkinson's diseases. One common age-related disease is osteoporosis, characterized by brittle and weak bones that make patients more prone to fractures and deadly falls. Using an X-ray imaging technique called micro-CT, the researchers found evidence for the reversal of age-related bone loss. Namely, while the aged monkeys treated with saline exhibited dental bone loss, the aged monkeys treated with SRCs had teeth more similar to those of young monkeys. The researchers also observed structural changes to various organs and tissues from aged monkeys treated with SRCs. For example, the vascularity of the lung and heart was improved, while the thickening of the aorta was reduced. Neurons had longer projections and fewer proteins associated with Alzheimer's disease, and the kidney and brain showed less mineralization. When it comes to combating degenerative aging, it makes sense that regenerative stem cells are a promising solution. In fact, one of the underlying causes of aging is stem cell exhaustion, whereby stem cells lose their regenerative capacity. From the improved memory to MRI scans revealing brain connectivity being restored and reducing shrinkage, rejuvenated organs and tissues showcases that SRC treatment helps slow down biologically aging. As no serious safety concerns were raised during the study, it would seem that SRCs are well tolerated. However, the long-term effects of the SRC treatment will need further evaluation. The primary concern with injecting stem cells into the bloodstream is that they can trigger the spread of cancer almost anywhere in the body. Nevertheless, the SRCs possess tumor suppression properties, suggesting they may not induce tumor growth. If this ends up being true, we may soon see SRCs being tested in humans. If this treatment became available, would you take it? Let me know in the comments below. I am actually pretty curious.